Welcome to Save Your Life with Joe, a channel dedicated to protecting and extending your life using science, logic, and technology. Today, we are going to take a quick deep dive into one of the most fascinating new precision medicine technologies, genomic liquid biopsy for multi-cancer detection. Now, we have heard about the cases of Claire, Jack, and many others whose cancers were detected at early stages using a new technology, genomic liquid biopsy. How can a simple blood draw detect cancer signals across all cancer types so early and small? Not even advanced imaging can detect them. And how are these different from other blood tests that we use to investigate potential cancers like PSA tests, prostate-specific antigen? Welcome to a quick deep dive into one of the most fascinating and powerful new medical precision technologies. This is about artificial intelligence, deep genomics, and single molecules that are being counted in the cloud. Let's start with some tumor basics. A cancerous tumor is an amorphous collection of cells that have gone rogue. It uncontrollably replicates and its cells have mutated. Their DNA has changed. Central steering mechanisms have been destroyed that normally control healthy cells and prevent these healthy cells from replicating uncontrollably. But tumors are not made out of super cells that cannot be defeated. They're actually super mutated, weak, and unstable. That means that a significant percentage of a tumor's cells dies every day. The bigger the tumor, the more cancer cells die at each moment in time, and their remains are transported out of the lymph system and the bloodstream. Part of these remains are fragments from their DNA. Yes, that DNA that contains the specific mutations that make a cancer, well, cancer. That is the lever genomic scientists use to detect cancers much earlier than previously possible. Using a blood sample, we can extract cell-free DNA, the DNA from dead cells that circulates freely in the blood plasma, and start looking for suspicious mutations. But doing so requires a lot of technology, like a lot. Okay, before we go into deep sequencing technology and AI, don't forget that you can always ask me questions down below in the comments about cancer, precision medicine technologies, DNA, and everything else we discuss here. I promise you to get back to every comment that makes sense, so let's get back to the deep tech. Sequencing is a technology that allows us to look at your DNA one nucleotide at a time. Nucleotides are the 4 different letters, ATCG, that make up the DNA. The problem is regular sequencing just randomly looks at each location a few times and creates the average of findings. So when it says you are an ATTG at this specific location of your DNA, what they really are saying is, I looked on average 30 times at these 4 locations and ATTG are the most prevalent findings. Actually, the A might be in fact 29 A's and 1 T, so we just say it's an A. That works because your genes are, well, your genes. They are mostly the same in all your cells. So knowing the approximate average is normally enough. But in cancer detection, it's quite different. Thousands of copies of cell-free DNA, to be accurate, roughly 2,000 copies in a 10 milliliter blood tube circulate in the blood. And only a very few, in extreme cases only 1, 2, or 3 molecules of this DNA actually stem from a tumor. There's always a lot of background noise. Even at late-stage tumors that have spread across the body, it's rare to see more than 2 or 3% of DNA in a plasma from tumors. And at early stages, we are talking 1 or 2 in 1,000. So to capture these needles in a haystack requires a massive leap forward in sequencing. To detect cancer in a single blood sample, up to 10 billion individual nucleotide reads are necessary. That's billion with a B. For comparison, a book of 80,000 words has about 500,000 letters. That's a full book. You're dealing with 500,000 letters. In other words, each time a precision liquid biopsy is performed on you, the equivalent data of 20,000 books is generated and needs to be read by someone. Each sequence has to be mapped to the genome to locate it and accurately identify potential mutations or variants. As you can imagine, this takes a lot of computing power, statistical learning, and artificial intelligence tools. That's why advanced liquid biopsy companies like Quantin are cloud and intelligence-first organizations. They need to have very sophisticated bioinformatics and cloud systems in place to process these 20,000 books worth of data for every single sample and every single patient. There's also a significant error problem in that some of the reads from the sequencing systems are wrong, and it's up to the genomics cloud to figure out which ones. Even a 1 in a million error rate would lead to 10,000 errors in 10 billion reads. And that's pretty bad if you have to be able to detect a single mutation on a piece of tumor DNA in the sample. Finally, the complex laboratory reports that reveal potential mutations in a sample have to be converted into medical intelligence and interpretation of what all that stuff means for a patient. That includes an assessment of how relevant a mutation is that is being found and how much of a cancer signal it actually carries. For that, the mutation frequency has to be compared with other patient samples who are either non-diagnosed controls or cancer patients. So the physician can get a clear picture if the level of cancer-associated mutations in a patient's sample is normal or abnormal. Since this is a new science and technology, this data has to be provided by the company that actually does these testings. Medical intelligence also takes into consideration other orthogonal factors. Orthogonal means 90 degrees, independent factors from the liquid biopsy, such as the patient's personal and family risk for cancer, that could be genetic based, and other screening results, for example, from full body or other imaging procedures. Liquid biopsy reports that simply say negative or positive should be viewed with skepticism. The science and evidence to do so is simply not there yet. And if anyone offers a test like this, the odds are they're pushing the envelope and not in a good way. Watch out for the following when looking at liquid biopsies. Number one, make sure you're not falling for a liquid biopsy that is not genomics based. Blood tests that simply investigate protein concentrations are thousands, some argue millions of times, less precise than genomics-based tests because they cannot count molecules, and the molecules they see are not unique to a tumor. They are more useful in downstream diagnostic scenarios when cancer signals are much stronger than in early detection and we are just monitoring a specific cancer. Second, make sure the liquid biopsy you get is either somatic variant based or a combination of somatic and methylation signals. Liquid biopsy that are only methylation based might be much cheaper, but methylation targets have much weaker backing in scientific literature, less understood, and don't do deep sequencing, so their results cannot be used in downstream oncology. They can supplement somatic variant investigation, but should not replace it. Reports that you get, that's number 3, should provide high-resolution insights into variant allele frequencies found. Right? They should tell you what is found at what frequency. They should provide comparisons for control cohorts so you and your doctor can see how you compare. They should not just give a simplistic yes or no answer. The technology is too complex and too early for that. A high-quality liquid biopsy has to always also provide medical advisory from a specialized precision medicine expert, both to you and your doctor. Every high-quality liquid biopsy always takes into consideration your personal and family history, as well as your germline genetics. And ideally, it is paired with other precision medicine modalities like full-body MRIs and other imaging. Now, liquid biopsy is becoming mainstream, with companies like Quantgene and Get Serenity, who also supports this channel, paving the path into a future in which all cancers are detected early and cancer is finally removed from the top 10 list of deadly diseases. That's the big vision. But it still requires a lot of expert insight and interpretation and is not ready yet to take your blood and just send you a yes/no answer. It's extremely exciting if we use it the right way. Now, these cases that we heard about and these new technologies are just another reason to take preventive medicine very seriously. Please use the comments below to ask me any questions you have about these technologies, about any criticism or ideas you have, but also about cancer, other preventive screening modalities like MRI and genomics. Don't be shy. It's your chance to get a straight answer to your questions. I promise I will respond. Please just don't troll me. If you have questions you don't want to share in public, you can reach me at this email here, saveyourlifewithjoe@gmail.com. That's my personal email for this channel. Everything stays confidential, of course, that you send, and I'm happy to help connect you to more resources or experts and point you in the right direction. You can also visit my private website, joebuckley.com, to learn more about free solutions that help you with preventative medicine and understanding your care gaps where you are late to screenings. It also helps you find precision screenings like liquid biopsy and full body MRIs in your own city. Bringing advanced early detection to everyone is our single best shot to defeating cancer as a deadly disease. We have developed precision screening technologies for billions of dollars in this society. Now, if you want to see liquid biopsy in action for early cancer detection and see how Howard, a 49-year-old real estate developer, detects prostate cancer when no one expects it and turns it all around, then click right here and watch the case. It's quite thrilling, and I really enjoyed that case. See you in the next video. This content is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of a board-certified physician or other qualified healthcare provider with any questions you may have regarding a medical condition. You should not disregard or delay in obtaining medical advice for any medical condition you have or think you have, and you should seek the assistance of a healthcare professional for any such condition. The views and opinions expressed in this podcast are my personal views and opinions and do not necessarily reflect the official policy or position of any company I'm involved in.